Hello, everyone, and welcome to our first online module. This week, we are going to cover biomolecules. So these are biological molecules within your body that have a specific function. So today, we're going to cover small and large biomolecules. Specifically, we're going to talk about carbohydrates, proteins, lipids, and nucleic acids. So let's get started. My goal this semester is not for you to get stuck in the weeds, so to speak, rather to take a step back from the cell level and try to see the big picture application of what we're learning. So today we are discussing biomolecules. These are found in the food we eat: carbohydrates, lipids, proteins, and nucleic acids. And as we know, and as you will learn, metabolism is central to body functioning and is the balance between catabolism, the breakdown of molecules, and anabolism, which is the building up of molecules. And this is known as metabolism. And it really comes down to the supply-demand relationship. How much do we need based on the demand of the body to produce energy? So energy production is required for all processes of life, from the cell to the big picture. Differences in energy production exist between foods. So we'll talk about this, that there's something called the glycemic index. There's different energy yield from different foods we eat, and they have a systematic impact. So biochemistry is central to nutritional guidelines, the specificity of exercise prescription from energy system design, as well as all of physiology is dependent on how quickly we can produce energy and then break it down to release the energy needed for human movement. So when we're looking at the cell, I want you to always try to go back to higher levels of the onion, so to speak, and see the big picture application of what we're learning. And this is really central to the focus of biochemistry. It's all about energy production and metabolism. Now let's firstly discuss different sizes of biomolecules. There are small biomolecules, and these are in their simplest form. The biomolecules are sometimes found in single units known as monomers. This means 1 unit. So this might be 1 unit of a fat, 1 unit of a carbohydrate. So a monomer is the smallest possible unit that still retains the characteristics of the molecule. So small biomolecules fall within an arbitrary range. We have monomers, which only have 1 unit, mono meaning 1, a dimer, di meaning 2, so 2 units, a trimer, meaning 3, so that would have 3 units, and an obligomer, which means a few. So a obligomer is anywhere between 4 to 50 units of that specific characteristic of the molecule. An example of a monomer for a carbohydrate would be glucose, also fructose and galactose. Then for a protein, the smallest form of a protein is an amino acid. And then 2 amino acids together would be called a dimer. 3 amino acids together would be called a trimer. The picture on the screen of the amino acid serine is 1 monomer. So serine is an amino acid. That's 1 monomer. That's the smallest molecule of that protein. And there's many different types of amino acids. It is possible to attach many monomers together. So this means a bigger molecule, which is more complex, and the biomolecules can exist as polymers, poly meaning many. So this means beyond 50 units. So typically polymers contain hundreds to thousands of monomers. This means for carbohydrates that you could have hundreds to thousands of glucose molecules bound together. Okay, polymers can be long linear chains in a row, or they could be branched chains or complex folded shapes. An example of this is alcohol dehydrogenase, which is known as an enzyme. And you can see the picture on the slide that this is actually made up of many, many, many complexly folded amino acids. Okay, so an enzyme is a protein that's made of many, many amino acids bound together in a very complex branched form. Now let's begin by discussing our first biomolecule, and this is carbohydrates. So as the word says, it's hydrates of carbon. So hydrates meaning H2O, hydrogen and oxygen, and carbon molecules. So we know that carbohydrates contain carbon, hydrogen, and oxygen molecules. That's it. Carbon, hydrogen, and oxygen. And so the abbreviation for carbohydrates is CHO. Now, when it comes to carbohydrates, we have small carbohydrates and we have large carbohydrates. The small are known as monosaccharides. Saccharides means sugar. So monosaccharides means one sugar. So the monosaccharides that are found in carbohydrates are 5, and I want you to write these down. The first is glucose. The second is fructose. The next is galactose. Then we have ribose and deoxyribose. Then we have disaccharides, or 2 sugars. This is when we have 2 monosaccharides bound together by a bond. And now we have 2 sugars together. The disaccharides that are formed are also known as carbohydrate dimers because there's 2 units bound together. Disaccharides can be called lactose, maltose, and sucrose. And we'll talk about those in more detail on the forthcoming slides. Essentially, lactose is a combination of a glucose molecule and a galactose molecule bound together, and that makes up the disaccharide known as lactose. We also have polysaccharides. Remember, these are many monomers bound together. Okay, so polysaccharides are carbohydrate polymers. There's many sugars bound together. And the ones that we have in the body are called amylose, amylopectin, glycogen, and fiber. Some of these are found in the body, and some of these are plant-based, large-scale carbohydrates. Okay, so we have amylose, amylopectin, which are both plant starches. Then we have glycogen, which is an animal starch, and fiber. And those are our 4 main polysaccharides. So it is really important for you to be able to name all of the monosaccharides, all of the disaccharides, and all of the polysaccharides that we just went over. So I want you to take a minute right now and try to name them. Now let's discuss our carbohydrate monomers. Let's start with glucose. So the chemical formula for glucose is C6H12O6. Remember, because it's a carbohydrate, it has hydrates of carbon. It's going to have water and it's going to have carbon. So we know that in this molecule specifically, there's 6 carbons, C6, there's 12 hydrogens, H12, and there's 6 oxygens. And the chemical structure is on the right. So C6H12O6 is known as the chemical formula. So this is a product of photosynthesis in plants, and it's a reactant of cellular respiration within animals as well as in the body. So for us, we require glucose in our diet to be able to break it down and to utilize it to produce ATP, our energy currency within the cell. So it's a very common energy source in most organisms, from anything from bacteria to humans. It tastes sweet, but it's not overly sweet. When we are counting the carbons within the actual chemical structure, we can see that on the far right side, we have carbon number 1, then it links with carbon number 2, which links with carbon number 3, which links with carbon number 4, linking to carbon number 5, and at the very top, we have carbon number 6. And we can see that each of the main carbons that are numbered have a hydrogen attached to it, bound to it, as a single hydrogen. And we also see that that carbon is bound to a hydroxyl group, an OH, also known as an alcohol. So the OH is known as an alcohol or a hydroxyl group. It's very important, the placement of the hydroxyl group, specifically for carbon number 4. Carbon 4 has the hydroxyl group, also known as the alcohol, facing or pointing downwards. This is known as an L conformation. So it has the OH facing downwards. Okay, this is important because galactose looks exactly the same as glucose, except its hydroxyl group is pointing up. And that's the only way that you can tell the difference between the two monomers. So it's extremely important to understand that so you can recognize which molecule it is. So again, recognize how you count the carbons. You go from right to left, C1 bound to C2, to C3, to C4, to carbon 5, to carbon 6. Each carbon has at least one hydrogen bound to it, and it also has the hydroxyl group or the OH group known as alcohol or hydroxyl. Very important to understand that. With glucose on the carbon number 4, we have the hydroxyl group pointing downwards. Now let's move on to discussing our second carbohydrate monomer. This is galactose. So the chemical formula is exactly the same as glucose. So C6H12O6. It's just that the chemical structure is different, as you can see in the picture. This is very important. So the chemical formula is the same as glucose. The chemical structure is different. And the only difference in the structure is at carbon 4, where the hydroxyl group or the alcohol is pointing upwards. This is the only thing that discriminates between the two monomers. That glucose is pointing downwards. The hydroxyl group is pointing downwards on carbon 4. Meanwhile, on galactose, on carbon 4, the hydroxyl group is pointing upwards. This makes a massive difference in the body, and it makes a huge difference to the type of monomer that you have. So this galactose is found in dairy products, sugar beets, and some fibers. It's usually not found free in nature, unlike glucose and fructose, but rather it's combined with glucose in the form of lactose, which is a disaccharide. Okay, so remember the disaccharide has two sugars. It has glucose and galactose bound together to make lactose. The C4 hydroxyl group is positioned upwards, and therefore it's known as galactose and not glucose. It's very important. That's the only difference between glucose and galactose. However, their biological function is completely different. You need to be able to recognize the difference between glucose and galactose, and so you have to be able to identify where carbon 4 is and the position of the hydroxyl group on that fourth carbon. If it's pointing upwards, it's galactose. If it's pointing downwards, then it's known as glucose. Now moving on to our third carbohydrate monomer. Remember, a monomer is the smallest unit of a carbohydrate, and this one's known as fructose. And I'm sure you've heard about this a lot. When you look at the ingredients in certain foods, you can see that fructose is an ingredient sometimes. So the chemical formula for fructose is exactly the same as galactose and glucose. So it's C6, 6 carbons, H12, 12 hydrogens, and 6 oxygens. So C6H12O6. This, however, the chemical formula is the same, but the chemical structure is different. In this case, let's look at the picture here. It's known as a 5-atom ring. So we have our first carbon way up on the top of the right side. So we have 1 carbon, 2 hydrogens bound to a hydroxyl group. That's carbon 1. Then you move from right to left. Carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6 is over on the other side, outside of the 5-atom ring. Now at the peak of the ring is oxygen. Okay, so within the actual ring itself, we have 4 carbons and we have 1 oxygen. So fructose is known as the fruit sugar. However, it's also found in honey and some root vegetables. It tastes sweeter than glucose, and it's used in combination with glucose as in commercially sweetening certain beverages. So if you look at Gatorade, it's usually a combination of glucose and fructose because it actually tastes sweeter. Overconsumption of fructose is actually not a good thing, that it actually is linked to obesity and diabetes. And we'll talk about this more on the forthcoming slides. So here, the carbons in fructose are numbered from the top right, number 1, to the top left. So with fructose, you actually get less of an insulin response compared to glucose, and that's why it can be problematic. However, too much glucose is also bad too because you get too much of an insulin response. But essentially these three, glucose, galactose, and fructose, all share the same chemical formula. It's just their structure is different, and it's important to know the differences between them and be able to identify which molecule it is. There are also 2 additional carbohydrate monomers. So there's 5 in total that we're going to talk about. So we've talked about glucose, galactose, and fructose. Now I'm going to introduce ribose and deoxyribose. So let's start with ribose on the left. Its chemical formula is C5H10O5. That means it has 5 carbons, 10 hydrogens, and 5 oxygens. It's found in RNA, ribose nucleic acid. It's highly concentrated in brewer's yeast. And you can see as well, it's a 5-atom ring, similar to fructose. The only difference is that you don't have 6 carbons like fructose does. Instead, we have 5 carbons. So you can see, again, you number the carbons from right to left. So carbon 1 has a hydrogen and a hydroxyl group. Carbon 2 has a hydrogen and a hydroxyl group. The hydroxyl group on the second carbon is pointing downwards. So we see that it's downwards, okay? And it's highlighted there in red. And then C3 is the same. C4 has a carbon bound to it, okay? Deoxyribose, its chemical formula is C5H10O4. So the difference between ribose and deoxyribose is exactly found in the name. Deoxy means you're removing an oxygen from ribose. So the name says it. Deoxyribose means you're going to have 1 less oxygen in its chemical formula. So the chemical formula for deoxyribose is C5H10O4. You've removed 1 oxygen from ribose. And this is found in DNA. So your DNA, deoxyribose nucleic acid, is naturally occurs much lower than that of ribose. Okay, so the structure of DNA is C5H10O4. Now, where does the difference lie? Where are you removing the oxygen? Well, let's look at the first carbon. We see a hydroxyl group and a hydrogen, similar to ribose. But then when we move to the carbon 2, we can see that the oxygen, we no longer have a hydroxyl group. We see that the oxygen has been removed from the hydrogen. And now we have a carbon that has a hydrogen bound upwards and a hydrogen bound downwards. So the site of removal of the oxygen is found on the second carbon. So look at the picture of ribose and look at the picture of 2-deoxyribose. The 2 indicates that the oxygen is removed from the second carbon. That's important to know. So the oxygen is removed from the second carbon. So it's 2-deoxyribose. Now you've been introduced to all of our carbohydrate monomers. We have glucose, fructose, galactose, ribose, and deoxyribose. Glucose, galactose, and fructose share the same chemical formula, but have different chemical structures. Ribose and deoxyribose are very close in their structure. However, one of them is missing an oxygen at the second carbon, and this is deoxyribose. You need to understand the properties of each and be able to identify them if the molecule is shown to you on a test. Now we're going to learn about our carbohydrate dimers, specifically the first one, sucrose. So remember, dimer means two of something, 2 units. So in this case, we have a glucose and a fructose binding together to form the sugar known as sucrose, which is very common. This is called table sugar, and it's also called sucrose. It's very sweet, it's comparable to fructose, and it's considered to be the most common food sweetener. It's important sugar found in plants, specifically pineapple and apricots. And it undergoes, when it's formed, when glucose and fructose come together, it undergoes something called a condensation reaction, where a condensation reaction is you're removing water. So you're removing a hydrogen and a hydroxyl group. So you're removing one hydrogen and a hydroxyl group, an OH. And in this case, it's known as water, H2O. So when you remove water, you actually have a bond that forms between the two molecules. And you can see it there. It's highlighted in red, and it's known as a glycosidic bond. And this is a bond. Two bonds are forming and sharing an oxygen group. Okay? So that's called a glycosidic bond, and it's formed through a condensation reaction. You need to know that. So dimers are formed through, specifically sucrose, through an interaction of glucose and fructose together, where we have a condensation reaction where we are removing water and we're forming a glycosidic bond. So how do we know which two molecules these are? Well, it's important to count the carbons. So first of all, we see a pentose ring over on the right-hand side. We see a 5-carbon ring. And in this case, we can count the carbons and we know there's 6 carbons. So in this case, we've ruled out ribose and deoxyribose because they only have 5 carbons. If we see that a molecule has 6 carbons and it's in a 5-atom ring, we know it's going to be fructose. On the left side of the molecules, we see, we can count the carbons, so start from the right and move to the left. We see carbon 1, carbon 2, carbon 3, and carbon 4. The hydroxyl group on the 4th carbon is pointing downwards. So in this case, we know that a downward pointing hydroxyl group on the 4th carbon means that it's glucose and not galactose. So we know that this molecule is glucose bound to fructose to make sucrose. The chemical formula is C12H22 and 11 oxygens, O11, because we've added the 2 sugars together. Okay, the 2 sugars are formed through a condensation reaction where we are removing water and forming a glycosidic bond where they're sharing an oxygen molecule. Now let's talk about our second carbohydrate dimer that's very common, and this is lactose. So remember, a dimer is formed when 2 monomers are bound together to form a dimer. Okay, in this case, for lactose, we have galactose and glucose being bound together. So the chemical formula for lactose is the same as for sucrose, so C12H22O11. What differs is the chemical structure, as you can see in the picture, which we'll talk about in a minute. So lactose is known as the milk sugar, and it makes up 2 to 8% of the solids found in milk. It's formed when galactose and glucose react together through a condensation reaction where we are removing a hydroxyl group and a hydrogen, so removing water, and forming a glycosidic bond between the 2 molecules. So the bond between glucose and galactose can be broken by something known as the lactase enzyme. But if you are lactose intolerant, you have a deficiency in the lactase enzyme, and therefore you can't break down in the process of digestion lactose. And so this leads to lactose intolerance. You cannot break it down and can cause havoc to your GI system. So similar to sucrose, we have 2 monomers coming and reacting together. In this case, we have galactose and glucose being bound together. So water is being removed through a condensation reaction. So we have 2 hydrogens and an oxygen being removed. And then we have a bond being formed between the 2 molecules. and they're both sharing an oxygen. And this is known as a glycosidic bond. So the glycosidic bond is formed through a condensation reaction where you're removing water. Okay, that's the extent of what you need to know. So how do we identify which molecules these are? Well, remember, it's all about carbon 4. So let's look at the molecule on the right. We start from the far right and we count the carbons. Whenever you see an inflection or a change in direction in the structure, we know that there's a carbon there. So we have carbon 1, carbon 2, carbon 3, carbon 4. The carbon 4 on the right molecule has interacted with the other molecule. And we know that the hydroxyl group was pointing downwards. So this would be called glucose. Now on the left-hand side, we can see our carbon 1, carbon 2, carbon 3, and carbon 4. And the hydroxyl group on the left-hand molecule is highlighted in yellow. And it's pointing upwards. So what do we know about that? We know that that is galactose. So in this case, we have galactose reacting with glucose to form lactose. Okay, so it's a combination. Remember, two monomers come together to form a dimer or a disaccharide, two sugars together. It's formed through a condensation reaction where we are removing water and forming a glycosidic bond where the two molecules are sharing an oxygen. Now let's talk about our last carbohydrate dimer or disaccharide. There's two sugars bound together. Remember, a dimer is when two monomers, the smallest units, are bound together to form a dimer. When it comes to carbohydrates, we have two monosaccharides being bound together to form a disaccharide. This one's called maltose, and the chemical formula is exactly the same as sucrose and lactose, and it's C12H22O11. The only thing that differs is the chemical structure, which we'll look at in a few minutes. So this is known as the beer sugar or the malt sugar. It's produced as part of the process of brewing grains, and it's the least common disaccharide in nature. Okay, and it's formed when glucose and glucose bind together to form maltose. So as you can see, how do we identify that this is a glucose? Well, we count our carbons. We start at carbon number 1, carbon 1, carbon 2, carbon 3, and carbon 4. And we can see that the hydroxyl group on carbon 4 is pointing downwards. This is highlighted in purple. Based on the rules we learned, the carbon 4, if the hydroxyl is pointing downwards, we know it's glucose and not galactose. So here we have glucose on the left, and on the right, how do we know that this is glucose and not galactose? Well, again, we count from C1, carbon 1, to carbon 2, to carbon 3, to carbon 4. We can see that the the hydrogen group is pointing upwards, and therefore we know that the hydroxyl group was pointing downwards. And in this case, based on our rules, if on the fourth carbon, the hydroxyl group is pointing downwards, we know it's glucose. So in this case, this molecule is maltose because we have two glucose molecules interacting together. This again, they're formed through a condensation reaction where we are removing water, and now we have a bond between the two molecules sharing an oxygen, and this is known as the glycosidic bond. So now I want to come back and discuss sugar a bit, specifically fructose. And fructose is known as the alcohol without the buzz. It's found in table sugar in the form of sucrose, as we just learned. Sucrose, remember, is 50% fructose because it's a combination of glucose and fructose. So why is fructose so bad? So you may have heard of high fructose corn syrup and things laced with fructose. Well, one, it's extremely cheap to use instead of glucose. And it provides more sweetening, so people will eat more of it. So why is fructose so bad? Well, fructose is only metabolized in the liver, and it gets metabolized and formed into glucose. and then used by the rest of the body. However, excessive consumption creates a fatty liver, which in turn leads to insulin resistance. And many researchers believe fructose is responsible for many of the diseases present today: type 2 diabetes, cardiovascular disease, and obesity. So what it does is it interferes with a hormone called leptin, and it interferes with your feelings of satisfaction when you eat. So what happens is you overconsume. So you don't have that feeling of satiety. So you eat more and more, and it impairs fat regulation. So before the mass production of refined sugar, people rarely consumed fructose, except for in fruit and in small amounts. Some people, as well, do not absorb all the fructose they consume, and this leads to something called fructose malabsorption. Fructose acts as a fermented carbohydrate, which can lead to excessive gas and GI discomfort. As such, it's characterized as something called a FODMAP sugar. And if you're interested in nutrition, you should look up the FODMAPs, FODMAP sugar, that many people misinterpret the symptoms of FODMAPs as being from wheat or gluten sensitivity or intolerance. But instead, you may have a sensitivity to fructose-containing foods. And as such, you should limit foods that have fructose in it. This is really important. And I truly believe that this is one of the reasons why people overconsume certain sugars and why we do have diseases present today. So try to look at the ingredient labels of the things you're eating, and avoid foods that have high fructose corn syrup, anything that has excessive fructose in it, because you'll just eat more of it. It tastes more palatable. It'll taste more sweet, and you'll want to eat more, but it leads to a fatty liver and a lot of issues later on. I now want to bring this real life for you. So let's look at some products that have these types of monomers, monosaccharides, and disaccharides within them. So this slide is kind of covered up, but on your slide you can see that I have two images here. One is Gatorade and the other one is a GU energy gel. So let's look at the ingredients of Gatorade. We see water, sugar, dextrose, citric acid, and a whole bunch of other electrolytes. Okay, so we know what sugar is. They just say sugar, but this is typically glucose, or table sugar, sucrose, which is fructose and glucose. And now what is dextrose? Well, really, it's a fancy word of saying glucose. It's just, you're not saying another sugar, essentially. So it hides it from the consumer. So dextrose is C6H12O6. It's identical to glucose, but it's synthetically created from corn. So it's not even a real sugar. It's using corn as a alternative to make it. And then we also see on the GU, we see maltodextrin. Well, maltodextrin is synthetically derived glucose polymer, so a polysaccharide. It's a food additive from plant starch, specifically corn. So many of these things found in these beverages and sport energy gels are from corn. And it's similar to be absorbed like glucose. However, it can irritate the GI system because it is made from corn, which is extremely cheap, and there's lots of corn in the world. So this is one of the reasons when I raced as a pro cyclist, I would consume huge amounts of GU. And because I'm sponsored by this GU product. So I had these gels in the back of my jersey, and I would take them. I'd set an alarm on my watch to take them every 30 minutes so I didn't bonk at the end of the race. And so, when you're exercising, glucose is your main energy source. It's broken down right away. It provides immediate energy. But after taking 5 or 6 of these in a race and then having them multiple days in a row, I started to get it's coined as gut rot. And it's really because you're consuming a high amount of artificial sugars, specifically corn. Like if you want to take sugar, you might as well just take the sugar, not an artificial sugar. But these products are laced with artificial sugars because they're cheaper to make. Instead, I decided to do some real food alternatives that carry phytonutrients and have higher bioavailability in the gut. So for example, you could have a boiled potato, you could eat figs, which are real sugar, or a banana, which carries sugar, but it also has vitamins and minerals or phytonutrients, and it gives you that immediate energy. So there's really only a few things that you can control in your life: what you eat and how much exercise you get, how you sleep, water. This is from a biochemical standpoint, right? There's other factors to life besides this. But from a food standpoint, what you eat, how you exercise, the choices you make around sleep, water, micronutrients are essential. And you must get these from your diet. So it's really important to choose alternatives that are going to provide energy but give you the best bang for your buck. So instead of spending money on these products, consider taking real food alternatives where you're getting the minerals, the vitamins, the micronutrients, and the macronutrients together. Now we're going to discuss our carbohydrate polymers. Remember, polymer means many monomers bound together. And the first one we're going to talk about is amylose. It consists of repeating chains of glucose monomers only, and it can contain several thousand monomers of glucose. It has only linear regions, so there's no branching going on. It's all one after the other in a linear format. It makes up approximately 30% of plant starch, which is the storage form of energy in plants. So as you can see here, there's glycosidic bonds between the two glucose molecules. How do we identify that this is a glucose molecule? Well, we go to our fourth carbon in each of the glucose, and we see that the alcohol or the hydroxyl group is in the downward direction. And when on the C4 carbon, carbon number 4, we see the hydroxyl group or the alcohol group pointing downwards, we know this is glucose and not galactose. You must be able to identify that. Okay, so a question I have for you is, when would you create a carbohydrate polymer? Or when would a plant create a carbohydrate polymer? And it really comes down to whether you're in a fed state after a meal or you're in a fasted state when you haven't eaten for quite a few hours. And this will really dictate whether glucose in the food that you eat will be stored in a stored form or it will be used right away for energy. So this is the balance between catabolism and anabolism. And it really is dependent on your need for the glucose. So for example, if you're in a fasted state and then you have a food that's rich in plant starch, like celery or any type of plant, you're eating the amylose from that plant and you're breaking down that glucose into its smallest monomers. Now, if your body needs that glucose right away, it will use it as an energy source. But if it doesn't need it, then you would store it in our stored form of carbohydrate within the body, which we'll talk about on the next coming slides. Our second carbohydrate polymer is amylopectin, and it consists of repeating chains of glucose monomers only. So again, it's just glucose bound together over and over again. And it can contain several thousand monomers, so it's a big molecule. The key here, and the key difference between amylose and amylopectin, is that amylopectin has both linear and branched regions that are insoluble. And it makes up approximately 70% of plant starch. So whenever you think of starch, you should think of amylose and amylopectin. So amylose is 30%, and amylopectin would make up the 70% of the plant starch. Now, the thing that you need to recognize here is that all the glucose monomers are bound together by dehydration reactions or condensation reactions, where you are removing water and you're forming a glycosidic bond. As you can see here in amylopectin, we have carbon 1, carbon 4 linkages, similar to amylose, but we also have carbon 1 to carbon 6 linkages. And this is what makes amylopectin branched. It has carbon 1 to carbon 6 linkages. And you can see this in the picture. You can find the carbon 1 and you see the carbon 6 of another glucose, and that we see a bond there. That's why amylopectin has a branched region. So here are two polymers so far that we've discussed, amylose and amylopectin, which makes up plant starch. Amylose, as you can see in the picture, is linear. We have linear bonds of glucose monomers, many thousands of them. And the linkages are carbon 1 to carbon 4 linkages only. while amylopectin is branched. So that means it has carbon 1 to carbon 4 linkages, and it also has carbon 1 to carbon 6 linkages of the subsequent glucose molecule. So amylose on the left is linear, and it has strong hydrogen bonds, and it makes it a lot harder to digest. Meanwhile, amylopectin, because it is linear and has branches, it has weaker bonds, especially the branched bonds are weaker, and therefore amylopectin is easier to digest. So a plant starch with more amylose, which is harder to break down, will have a lower glycemic load as it takes longer to digest and release that glucose into the bloodstream. So we just talked about the two carbohydrate polymers that make up plants and plant starch. So when you take a plant or you take even a piece of celery or a piece of lettuce, it's composed of amylose and amylopectin. Now let's look at our animal starch, and this is known as glycogen. And you probably are more familiar with this carbohydrate polymer. Well, it also consists of repeating chains of glucose monomers only. So there's no fructose and there's no galactose. It is only glucose monomers. And it can contain several thousand monomers. It has both linear, so carbon 1,4 linkages, and branched regions of 1,6 linkages. This is known as 100% of animal starch, and we can store approximately 2,000 kilocalories of glycogen in our body at one time. So in the liver, we can store approximately 400 kilocalories of glycogen, and this is mostly for glucose homeostasis. So when you're sleeping, we still need to maintain your blood glucose level, and your liver glycogen helps in doing this. And then in our skeletal muscle, our contracting muscle, we have energy storage as well, and this is mostly to sustain the intensity of exercise. And we have a capacity of about 1,600 kilocalories of energy capacity within the skeletal muscle. So of the 1,600 in the skeletal muscle and the 400 capacity in the liver, we have a total of 2,000 kilocalories of glycogen being able to be stored within the body. So we have branching and linear. It's similar to amylopectin, but they're not the same. That glycogen has more relative branching than amylopectin, and that's the main difference. So what is the significance of being able to store glucose monomers in the body? Well, this is known as something called redundancy of control. We want to always be able to sustain our blood glucose level when we may be fasted for a long time. So it's almost like a survival mechanism that we have 2,000 extra kilocalories of glucose monomers within our body that we can break down and use to keep our blood glucose levels in a good state whereby our body is in homeostasis, specifically for our brain, because the brain requires glucose. Glucose is the only nutrient that can pass the blood-brain barrier. So your brain runs on glucose, on this monomer, and so we need it in our body to maintain the functionality of our brain. So here's a nice picture of glycogen. As you can see, it's quite different in appearance compared to amylopectin. We have more relative branching when it comes down to glycogen, so more 1,6 linkages. In the middle of glycogen, we have a protein core, and it's called glycogen. And then we have our individual glucosal units known as one glucose monomer, and you can see it that's circled in red. So this is a more realistic picture of the structure of glycogen. The center, again, is made up of that protein core, and we only access the carbohydrate, the glucosal units, the glucose monomers for energy. We never get to that protein core. So if we're in a fed state, so this is after we just ate a meal, we will have excess glucose within our blood, and this will be stored. So we'll use what we need, and then we'll store the rest. So this means we're forming glycosidic bonds between glucose monomers to create glycogen through condensation reactions. If we're in a fasted state, so we haven't, overnight, so you haven't ate while you're sleeping, and you woke up and you're going to have breakfast. Well, what happens during that so-called fasted state when you're sleeping? Well, we liberate some of the glucose monomers for energy production and blood glucose homeostasis. So what happens is you break the glycosidic bonds between the glucose monomers through something called hydrolysis reactions. And we'll talk about those reactions another time. But that's the difference. When you're fed, excess glucose monomers in the body get stored as glycogen. When you're in a fasted state, you break down the glycosidic bonds and release glucose into the blood to maintain blood glucose. So the fate of glucose within the body is very much dependent on your blood glucose concentration. That, for example, if we're in a fed state, typically our blood glucose will rise because we overload the body with glucose, whether it be from complex carbohydrates or more simple carbohydrates. We have an excess amount of glucose. So that excess glucose will be stored as glycogen. So we create glycosidic bonds between the glucose monomers and we store it as glycogen. Now, if we are fasted and we haven't ate for a while, even 4 or 5 or 6 hours between meals, that's a long time. And so we need to still maintain our blood glucose concentration because that blood glucose is circulated to the brain, to every organ within your body, and glucose is a main energy source, especially for the brain. It's the only nutrient that can pass the blood-brain barrier. So you need glucose. And so we have to maintain our blood glucose concentration. And so what we would do in a fasted state is we break down the glycogen. We break down the glycosidic bonds between the repeated glucose monomers through hydrolysis reactions where we add water and we break those bonds and then we liberate that glucose to be dumped into the blood and then we can maintain our blood glucose concentration. So it's a fine balance between catabolism and anabolism where we're breaking something down like glycogen or we're creating glycogen through anabolism. That concludes our online lecture for this week, where we talked about small versus large biomolecules, small meaning monomers, dimers, trimers, and oligomers, large meaning polymers, and the unique qualities of polymers. Then we discussed our carbohydrate monomers, where we defined 5 of them, and then we went into our carbohydrate polymers. Thanks so much, and see you next week.